Hi, I'm Park Ranger Joel Kane,
Hi, I'm Ranger Ronnie.
and welcome to yet another day of amazing talks here for the virtual Grand Canyon GeoFest 2020. Today's a very special day right in the middle of the week of GeoFest. It's National Fossil Day.
Yay! My favorite time of the year.
So, of course, in addition to some other awesome events going on today for National Fossil Day, our GeoFest talks today are focused on fossils and paleontology here at Grand Canyon.
Yeah.
So fossils are really amazing things. It's kind of a subfield within the field of geology. But as I like to say, well, you can't really study fossils without also studying the rocks that they're found in. So all paleontologists who study fossils are also, to some extent, geologists. And there are also quite a few geologists who also study fossils.
This is true. Fossils enrich our life, and the purpose of National Fossil Day is a celebration by the National Park Service that coordinates this nationwide celebration that really promotes the educational and scientific value of fossils. They are so unique. They are a snapshot in time that can be a clue to past environments.
Absolutely, we learn so much from fossils. We learn about what the environment was like when rocks were being deposited. We can learn about the passage of time, the evolution of life. So that's why we're focusing so much on them for today as part of GeoFest.
And they're fun.
Absolutely, yeah.
For us kids of all ages,
Of
right?
course, yeah. No, I mean, I fell in love with fossils the moment I saw my first dinosaur skeleton at the Smithsonian Natural History Museum in Washington, D.C. And, of course, oftentimes when we start talking about fossils here at Grand Canyon, one of the first questions people ask us is, are there dinosaurs
Yeah,
here at Grand
sadly,
Canyon?
no dinosaurs here. Our youngest rock that we're standing on right now is 270 million years old,
Yeah.
which was way before the dinosaurs
A little
were
bit
around.
older. A little bit before the dinosaurs. Yeah, so all of our rocks here are actually older than dinosaurs,
Yeah.
which, you know, is kind of crazy because we think about dinosaurs as being pretty old.
Yeah.
But all of our rocks are older. But we do have a lot of amazing other fossils that are found in all the different sedimentary layers within Grand Canyon. And our talks today are going to be telling us all about those really amazing fossils that we find out there.
Yeah, and that's one thing about Grand Canyon. About a third of Earth's history is actually exposed right there in those colorful layers that you see behind us. And each one of them represent an ancient ecosystem. There's ancient deserts, beaches, swamps, oceans, and they're all visible out there. And if you know the language of fossils and know the language of geology, you can kind of read those stories that's been preserved.
Yeah, absolutely. And of course, since the rock layers of Grand Canyon are so well exposed and so well preserved and still basically flat lying, you can start from down there towards the bottom of the canyon, work your way up layer by layer, and as you see how the fossils are changing in each layer as you go up, you're literally watching the evolution of life on Earth as different types of plants and animals die off, go extinct, other types of animals and plants replace them. So yeah, our first talk today is going to be focusing on actually fossils that you're not going to see with the naked eye if you go out there looking for them in Grand Canyon, because they're very tiny. They're microscopic. Our first speaker today is going to be telling us about a whole world that we don't actually see with our naked eye, a world of microorganisms that is preserved in some of the oldest sedimentary layers down there at the very bottom of the canyon. So I hope you enjoy that first talk here today for GeoFest National Fossil Day edition. So thanks for joining me here to introduce our talks today, Ranger Ronnie.
Oh, my pleasure. Any excuse to talk about fossils, I'm there. So happy National Fossil Day to all of you. Be sure to check the National Park Service web pages. There are events going on all throughout the country, and maybe there's some in your backyard.
All right, so like I said, our first talk today, microfossils. And some of the oldest layers in Grand Canyon, and then a couple other amazing talks after that, ending with our park paleontologist at the end of the day here. So thanks for joining us today for National Fossil Day and GeoFest 2020. All right. Well, hello, everyone, and welcome to our first talk for today. Today, of course, is National Fossil Day. Today's talks here is part of Grand Canyon Geology Festival, or GeoFest 2020. Today's talks are all about fossils and paleontology here at Grand Canyon. Here to talk to us in our first talk today about some of the earliest fossils that are found within Grand Canyon, some of the oldest sedimentary rocks down there at the very bottom of Grand Canyon, is Dr. Susanna Porter. She got her PhD in biology from Harvard University in 2002. And her PhD work largely involved work on some of these early Grand Canyon fossils, these microfossils in Grand Canyon. After getting her PhD, she did a postdoc for a year with NASA in astrobiology at UCLA. And then in 2003, she moved to the University of California, Santa Barbara as an assistant professor. And she's continued to work on these Grand Canyon microfossils and also on biomineralization, some of the earliest animal skeletons. Basically looking at the kind of the oldest records of early life we have, things like the Cambrian explosion. Dr. Porter, thank you very much for being with us here today.
Thank you, Joel. I'm very excited to be here.
All right.
And to kick this off, this Fossil Day celebration, to talk about some of the oldest fossils in the Grand Canyon. I'll get my slides up. Hopefully that looks okay. So as Joel mentioned, I did part of my PhD working on some of the oldest fossils in the Grand Canyon, and these come from a unit of rocks known as the Chuar Group. Some of the Chuar Group rocks are shown here in the foreground in this lovely photo from my colleague Carol Daler at Utah State University. The Chuar Group is part of a larger package of the oldest sedimentary rocks in the Grand Canyon, known as the Grand Canyon Supergroup. You can see this here in this diagram. These rocks overlie the metamorphic basement. Vishnu schist is shown here, and they underlie, under the Great Unconformity, Cambrian and younger age sedimentary rocks that are more flat-lying. You can see these here. They're exposed only in a small part of the Grand Canyon. I've shown it here in the eastern part, circled in yellow. Now, the fossils in these rocks provide a fascinating glimpse of what life was like 750 million years ago in a shallow, calm seaway located pretty close to the equator in a basin that formed as a result of the breaking apart of the supercontinent Rodinia, a reconstruction of which is shown here. Before I talk about these fossils, though, and show you pictures of them, I just briefly wanted to talk for a minute about give you an overview of the history of life, just so you have a sense of where they fit in the big picture. And to start, I wanted to briefly talk about what we know about the tree of life. I think one of the most amazing discoveries that in the last few decades is the incredible diversity of microbial life on Earth today. And I think this is illustrated really well by our changing views of the tree of life. So this is a tree that was put out in the late 1950s. It's actually the same tree that I learned when I was in high school in the 1980s in Seattle, Washington. And it shows the 5 major groups of life: the multicellular plants, the fungi, the animals, and then the single-celled groups, the protists, and the Monera, which we can think of as the bacteria. The bacteria. And this is the tree of life from published just a few years ago. And it's an unrooted tree, but what it shows is the vast genetic diversity of life resides in the bacteria. You're all familiar with the bacteria. They are cells that don't have a nucleus. They're fairly simple in their appearance. And then there's also this group of fairly simple cells known as the archaea, which is shown here, also lack a nucleus. And then eukaryotes, this group here, is a pretty small twig on the tree of life, and that's where we reside. The eukaryotes include the animals, plants, and fungi. You can see up here, shown in this picture. Also includes the seaweeds and the slime molds, but the vast majority of eukaryote diversity is single-celled, the protists. And these things are pretty underappreciated. They're morphologically and behaviorally often quite complex, and as you can see, they are stunningly beautiful. So the eukaryotes are here, a pretty small branch on this tree of life. And animals, our group, is such an insignificant twig that it's not even shown on this tree. It is part of this tiny group, the opisthecons, which also includes the fungi. So you would think that given that most of modern diversity of life is microbial, and that animals are just a small branch on the tree, that the appearance of animals would be quite late in the history of life. And in fact, that's what we see when we look at the fossil record. So here's a simplified history of life. The Hadean is a time period that by definition has no rocks. And then the first time we actually have sedimentary rocks that would be capable of preserving fossil evidence of life, we see fossil evidence of life. So we know life was around basically, almost as soon as the Earth got started, probably. And yes, indeed, multicellular, like animals in particular, are very late, appearing around 600 million years ago, and plants even after that. So most of the history of life on Earth is one of microbial organisms. Simple cells, so those bacteria and archaea, they diversify and spread across the globe, living in all sorts of environments by 2 billion years ago. And then beginning around 1.7, 1.6, we start to see evidence of these more complex cells, the eukaryotes, our ancestors, appearing and diversifying. Here's a picture of what some of these oldest eukaryotes look like. They are about 1/10 of a millimeter in diameter. You can see they have various ornamentation, some spines coming off of them, and that's why we think they probably are our eukaryotes, our complex celled ancestors, rather than bacteria. By about 1 billion years ago, we start to recognize what some of these fossils are. We can place them in different known groups within the eukaryotes. So we see evidence of what looks like red algae at about 1 billion and green algae at about 1 billion as well. So we start to see the diversification of the modern groups of eukaryotes beginning about 1 billion years ago. Now, where does the Grand Canyon fit? Well, I've shown you here in this blue bar the span of fossil-containing rocks in the Grand Canyon. Of course, there's much older metamorphic rocks in the Grand Canyon, but just to focus here on the fossil record of the Grand Canyon, you can see it crosses this time interval from before we have animals through to, we have plants and even invertebrates around. So the Chuar group, what I'm going to talk about today, occupies this lowest part of that blue bar, the time period when these complex cells have diversified or beginning to diversify before we have animals. So let's turn to the rocks themselves in the Chuar group and see what they look like. Here's a stratigraphic column here on the left showing that actually the Chuar group is quite well constrained in terms of its age. These are various radiometric age constraints, most recently done by Alan Rooney et al. Here's again a picture of these beautiful rocks. And I want to focus first on fossils that that we found in the uppermost part of the Chuar group, in the Awadabi and Walcott, and in particular, fossils that come from these fairly large carbonate nodules that formed after the deposition of these shales all around them. So the shales were deposited, and then through various processes related to probably decay of the fossils I'm going to show you, the carbonate was precipitated around in the pore spaces and resulted in these often quite large nodules. You can see a backpack down there. If you take one of these nodules, cut them up in thin section and look at them under a light microscope, what you find is stunning. These fossils are, these rocks are chock full of these fossils. And in fact, these nodules are pretty common at the upper part of the Chuar group. You can see they're just cheek by jowl in there in the rocks. There's probably 1 trillion of these microfossils in the upper part of the Chuar group. And these microfossils are beautiful. They're known as the vase-shaped microfossils because they look just like a flower vase with an opening at one end. You could stick the flowers in. And these in particular that I released from putting the rocks in acid and getting these fossils out are these beautiful silicious clear vases, like a glass vase. These look strikingly similar to a group of modern protists known as the archalinid amoebae, a group of testate or shelled amoebae. So these are, you're probably all familiar with the naked amoeba. This is an amoeba which makes its own test, makes its own shell, like a snail makes its own shell. Here are some pictures of living testate amoebae. You can see it lives in its little house, but it sticks its little pseudopods, its fingers outside to grab prey. And these archalinid amoebae make a variety of tests. A lot of them, they all make a sort of an organic type test, and some of them don't decorate it at all. It's just the organic. Some of them make their own secreted particles, commonly silica, and they glue them together on this organic test. Here's a picture of one with square. Here's another one with oval particles, mineralized particles. And then a number of them make tests out of they agglutinate. They take particles from the environment, maybe grains of mud, or in this case, the empty skeletons of other protists living in the environment. These are all modern examples. And this one has taken scales formed by its prey that it's eaten up, digested the test, and then it steals these little mineralized scales and builds its test out of it. You can see those pictures there. All right, so I studied these fossils as part of my PhD, and basically my PhD work was to show how similar they were to this modern group of archalinid testate amoebae. And I'm just going to show you a couple examples of the modern versus the fossil, where you can see they're pretty strikingly similar. So here is an example, fossil in yellow, modern in blue. Here's another species, Bombycian micron, which has this opening where the pseudopods come out, sort of slanted, almost like a face. Here's another where the shell is a hemisphere in shape and it has a circular opening or aperture, almost an exact sort of comparison to the modern Archaea. Here's another with a triangular opening, perfect triangular opening, similar to what we see in this modern group Trigonopixis. Here's another which has 6 indentations surrounding the mouth of the test. You can see here where it's looking straight on at the mouth of the test, where you get a hexagon, a circular opening inside, and various of these features are known today, also in the Archaea. There's another number of other examples too, but I don't have time to go into them. But the interesting takeaways here is this is one of the earliest examples we have in the fossil record where we can take a particular fossil and assign it to a branch in the modern tree of life. I mentioned the red algae and the green algae before, and this is another pretty compelling example that we have this group in the eukaryotic tree, the Amoebozoa, which contains amoebae, that they were around at this time. We can sort of put a mark there on the tree. Branching of the tree. The other interesting thing about these fossils is that we know from the modern groups that they are pretty voracious predators. They eat algae, they eat bacteria, they eat other protists, and they even eat animals. They live, they're common today in mosses and peat bogs, and you can see this is a series of different photos that show the trapping and consumption of this little animal known as the rotifer. So here we have some of the oldest evidence for predation for predators in the fossil record, these fossil organisms. I'll get back to that in a minute. So I want to turn now to some of the older rocks in the Chuar group. And you'll note in this stratigraphic column that much of the Chuar group is fine-grained rocks. It's shales and mudstones. That's all this white layer here in the stratigraphic column. And you can see pictures of this here. For the most part, these shales are chock full of microfossils. It really is a gift from the Grand Canyon, this beautiful fossil record. If we take some of these rocks and we stick them in hydrofluoric acid, dissolve the sediments away, what we find left over is organic residue that contains a whole diversity of lovely organic wild microfossils. They're flattened, as you can see in this picture here, because they've been smushed by the weight of the rocks over them, but we still get a sense of their ornamentation. I have, in recent years, started looking at these fossils using scanning electron microscopy, where I just put a little bit of that sludge on a slide. I look for these fossils in amongst sort of the gunk, and then find them under the microscope, and you can see all these little kind of circular flattened discs are fossils. So they're quite abundant. And here's some examples of a number of these beautiful fossils shown here. And the diversity, the taxonomy of that is a whole other story. But what I wanted to call your attention to is that this sort of surprise discovery that I made in the process of studying these microfossils under SEM. And that is, so I would look, so here's a beautiful fossil. It's been squished, but it's pretty well preserved. And what I started to notice looking at these up close is there would be these tiny, perfectly circular holes. Here's an example of one here. Here's another fossil. If you look up close, you can see it has all of these little holes, all about the same size. A tenth of a micrometer, which is, so it's a 10,000th of a millimeter, these holes. And we can cut through them and we can see that they're beveled. So they're sort of slanted inward. This is the inner part of the wall. That's the outer part. Here's another example. Yet another example of holes all about the same size. And finally, this example, larger hole, about a micron. And you can see this also, that sort of beveling. So my first thought is, well, you know, could these be pores in the wall? We know that there are some fossils that do have pores in the wall, but these are much more regularly distributed if they're actually a characteristic of the species. So that seems unlikely. Another possible explanation that I struggled with is, well, could they be formed by minerals that are growing after deposition and puncturing through the wall? Well, we can see what that looks like. This is an example of a fossil where you can see it kind of has an ugly surface, and it's part of it is a result of minerals that are growing into the wall, like pyrite. It looks much more irregular than these lovely holes. Another possibility is maybe we see microbial degradation. Maybe this is just bacteria that are eating the dead cell in the sediments. So I looked a little bit into the literature of what it looks like when bacteria are degrading, say, plant cell walls. And what we find is a very different pattern. The wall is sort of much more widely eroded, and you see different stages of decay. And what's striking about these holes in these fossils is that they're all the same size. We don't see different stages of decay in the degradation. Instead, what these holes look a lot are reminiscent of are holes made by predatory organisms known as vampire amoebae or vampirellids. So this is literature that goes back decades looking at predators on fungal spores and hyphae. You can see examples of these holes here. They're quite tiny, but they're perfectly circular. And this process has been studied, and what we know is that the amoeba, which is this net-like form here, captures the fungal spore and it erodes a circle in the wall, and then it lifts off the sort of inner disc, almost like a manhole cover in the street. It lifts it up, it ingests it, here's a part of that disc there, and then it inserts its pseudopods into the hole that it's formed, and it eats up the yummy stuff inside. Here's an example. You can see color photos of this vampire about to prey on this green alga. Here it has bust through the wall. You can see another example here, and it's basically either the trigger pressure, the guts come out of the cell, or the amoeba sticks its pseudopods in and sucks it up. And there's one that's infested. They're a real problem actually in ponds growing algae for biofuel, these predators. So I thought, oh great, here's another opportunity for me to identify another branch in the eukaryotic tree, the vampire amoebae. And the vampire amoebae are right here in this group of eukaryotes known as the Rhizaria, includes Radiolaria, Foraminifera, maybe some of you have heard that. The problem is, however, that the more I dug into the literature, I realized that this type of sort of piercing, sucking vampire type predation is quite common. It's spread throughout the tree of eukaryotes. It occurs in the amoebozoa. It occurs in the Opisthokonts. That's a group where the animals and fungi are, so some of our close relatives do this. And there's a couple other examples in the Rhizaria as well, among other non-vampirellid groups. And so this seems to be a pretty common feature that's spread throughout our relatives, the eukaryotes. When I was doing this work, it occurred to me, I recalled back when I was studying these vase-shaped microfossils in graduate school, that a number of them had these unusual holes. There's two types of holes, in fact. One that has this semicircular, one hole is semicircular, you can see here. They're all about the same size, and interestingly, they're all aligned with the sort of long, straight side of the semicircle, aligned with the long axis of the test. And then there are others that are actual circles. You can see here and here and here and here. If these are indeed holes made by predators, which I suspect they are, the fact that these semicircular holes are aligned in such a specific way, I think suggests some type of sort of relatively complex prey behavior, that they orient their prey in a particular way and then use this cutting technique to get at to cut out this semicircle. I have no examples in the modern of a semicircular hole, though, so this has remained a puzzle to me. All right, so this is a pretty brief talk, but I just wanted to sort of highlight what I think is a really fascinating glimpse of life at this time in the Chuar Sea. So we have a number of fossils, a couple dozen, maybe 3 dozen species of microorganisms, of protists that we see in the Chuar group. So we know life was incredibly diverse, but in addition, we see evidence for a lot of different predation, different kinds of microbial predators. So we have the drill holes in the vase-shaped microfossils, the semicircular ones, and then the circular ones, probably 2 different types of predators. And these are much larger than these very tiny, 1/10,000th of a millimeter holes that we see in these organic walled microfossils. And what we know from the moderns probably represents different species. And then the vase-shaped microfossils themselves, we know, are predators. So we have a situation where if we look at this quiet, calm ocean, it looks like nothing is going on. We don't see really any multicellular life. But in fact, we have this sort of predator-prey, fierce predation going on in the microbial realm. We just can't see it if we travel back in time, but it's there. So that's all I wanted to say, and I wanted to thank the organizers of this and Joel for help with videotaping, and I welcome any questions.
All right, awesome. Thank you very much, Dr. Porter, for that amazing talk, because it's a really interesting look at kind of a whole world that I think a lot of us as human beings don't really tend to think about. The fact that there's those figures of the tree of life you showed were kind of amazing to think about the world that we commonly think about, on our scale is just a twig on a branch of this whole huge tree of life. And there's all these crazy things happening at a microscopic level, constantly all the time. And that's all been going on for billions of years.
Yeah, and I think learn about these single-celled eukaryotes, this protists that are, our close relatives. We often see situations where they have solved the same problems that we, that our ancestors solved in terms of evolution long before we did, and often in sort of similar ways. So, the predation, one of the explanations for the Cambrian explosion is that oxygen levels got high enough that you could actually get organisms that were able to be predators, that had high enough metabolisms, so they could chase things around, and that predation really is sort of the proximate cause driving this great diversification of animals. When you think about it, and one of the things that animals evolved was armor. We see a lot of mineralized skeletons at this time, right? When you think about it, so we know that these single-celled organisms, these eukaryotes, were experiencing predation long before animals did. And in a lot of cases, they came up with the same solution. So I didn't have time to talk about it, but there are these beautiful mineralized scales that appear, this is described by Phoebe Cohen and others, And rocks about the same age, mineralized scales that have these spines coming out of them, and we think that they probably armored a single cell. And so,
Oh, wow.
which is a sort of indirect evidence for predation at this time, that they're, protecting themselves in the same way that animals figure out the solution. Same goes with eyes. There are single-celled organisms that seem to have what appears to be an image-forming eye. These are a group of dinoflagellates. And it's all made of subcellular components, but they have sort of the equivalent of a lens and a cornea and a retina. So anyway, these are just fascinating creatures. And I think, most of us, I didn't appreciate how amazing they are.
Absolutely. I mean, when it comes to fossils, things, well, one of the first things anytime we talk about fossils here at Grand Canyon that a lot of folks, especially younger folks, ask, it's like, well, are there dinosaurs? Are there dinosaur fossils? And I always have to say, well, no, there's not. Our rocks are too old. But it's things like dinosaurs that get so much of the attention. Because they're we can see them and they're on like kind of our scale and and we can imagine them kind of living in the world that we live in. Yeah. But there are whole other worlds, right here on our own planet of things that we can't see. And there are like what you've talked about. There's rocks in Grand Canyon that tell those stories. Yeah. And we're going to be hearing a little bit more later on about some larger fossils in the canyon, like fossil trackways, not from dinosaurs, but from from animals. But it's really, I think it's really amazing to start out our talks here with National Fossil Day with this whole other world made of these tiny little things in rocks way down there towards the bottom of the canyon. That a lot of folks might not have even really thought about.
Right. And I like your use of that word, sort of whole other worlds, because that's how I think about it too. That, when I was young, I wanted to be an astronaut that went to other planets and discovered alien life. I thought that would be amazing. And in fact, maybe that will be possible in the next few decades. Send astronauts to Mars and look for ancient life. But I realized as I got older that, the best possible way to see strange foreign worlds and foreign life, alien life, really strange life, is to be a paleontologist. And you sort of think about even going back to the dinosaur time. It's amazing to me every time every once in a while I have to stop myself and think, oh my God, because I teach a class on dinosaurs. I think, wow, these things actually existed. They're not just monsters in horror movies. It's incredible to think these creatures roamed roamed North America. And that was pretty recent. And you go even further back and there's strange tall trees, nothing like we have today, totally different groups. They're practically extinct today. And then of course, even further back, to the time I study, it's like an alien planet. I mean, it's our history, it's our ancestors. Yes. And yet it's such a different world. And it's like, it's time travel, but it's also like traveling into outer space in a way to alien worlds. It's just.
Absolutely. I think about it the same way sometimes too. When I think about past landscapes, past ecosystems, with things that we're living that aren't around anymore. It's, and that's kind of what we see when we look at all the different rocks in Grand Canyon. We're seeing all of those stories and all those ancient worlds that used to exist here on Earth, that are very, very different from what we know today. Yeah. And it never ceases to amaze me. So
yeah, and it's interesting. It makes you. I mean, I actually find it kind of comforting. It makes me feel so insignificant. Our species is so. Of course, we are significant, but in a way, so insignificant in the story of Earth and the story of life, which I find comforting.
Yeah.
And its vastness. Yeah.
Oh, I know what you mean. I sometimes go out along the rim of the canyon and I look out and I think about the vastness, not just in space, but in time and the amount of history that's contained, in rocks all over the world, but even just here in this one spot, in this one, part of the world. Yeah. And it blows me away when I really start thinking about it. So
We're so lucky to have it here, and yeah, so lucky we can just go visit it and look at it and touch these rocks.
Yeah, absolutely. No, and I wanted to be a paleontologist as a kid, well, and an astronaut. So I can empathize with that. I know exactly what you mean. I ended up studying geology in general, but I have a special interest in paleontology, as well. So.
Yeah, well, I mean, even geology. I tell my kids, you can pick up a rock, and a rock seems like such a, kind of a boring thing. Whatever, you throw it out. But this rock has an amazing history. I mean, every rock has an incredible story to tell in terms of how it was formed, how it got to where it is today, what the planet looked like at that time. It's so funny, something so simple that's everywhere, but if you stop and think about what it really means, it's mind blowing.
Yeah, I love what you said about it as being our history, our story. I mean, this is, you look at things like rocks, or you look at fossils that are up to like 1 billion years old, and you're like, why does any of that matter? Why do we care about these things? And it's like, that's our history. That's where we came from, and that's where the world we live in today, where it came from.
Yeah, exactly. To understand why things are the way they are today, you need to study your history.
Absolutely, yeah. Well, awesome. Thanks again so much for being with us here today, and for giving us a look at this whole other world that's preserved here at Grand Canyon. So. Yeah.
There's lots more to learn too, I should say. I feel like we're only scratching the surface here. So
yeah, definitely.
Future paleontologists.
Yeah, exactly. For those of you out there watching, future paleontologists. Yeah. So.
All right. Well,
all right. Thanks very much again.
Yep.
Well, hello again, everyone. I hope you enjoyed that first GeoFest talk for today. It's National Fossil Day, and we are celebrating, of course, with talks all about fossils and paleontology here at Grand Canyon. That first talk was about microfossils, this whole other world, this whole other web of life that once existed in some of the oldest sedimentary layers way down in the depths of Grand Canyon. That was, I thought, a pretty amazing talk. And our next talk is going to be focusing on fossils that are a little bit larger, but actually a different type of fossil, a different group of fossil called trace fossils. Now, instead of body fossils, which is the actual remains of living things, this talk is going to be focusing on traces left behind by living things. And whereas body fossils, the actual remains of something, can tell us a lot about what it looked like, trace fossils actually tell us about how a living thing behaved when it was alive, whether it crawled or slithered or swam across bottoms of shallow seas or across the landscape. So trace fossils can be really amazing. We learn a lot of really useful information from studying them. And before that next talk, I brought us out here again to Plateau Point. Some of you might remember that I ended the day yesterday out here at Plateau Point, and I decided to come back because, well, Plateau Point is amazing, and also because the Tapeats Sandstone, which is what I'm standing on, is my favorite rock formation in Grand Canyon. And there's some pretty amazing trace fossils in these layers of Tapeats Sandstone. So that's why I came back out here to Plateau Point, and I hope you appreciate that view back there behind me. And I hope you enjoy this next talk on trace fossils of Grand Canyon, celebrating National Fossil Day here for GeoFest 2020. Thanks for joining us.
Well, hello again, everybody, and welcome to another one of our talks here for today, National Fossil Day. Right here in the middle of Earth Science Week and Grand Canyon Geology Festival, or GeoFest 2020. So, of course, National Fossil Day, we're learning all about paleontology, all about fossils here today. And here to talk to us about fossils in Grand Canyon is once again Dr. James Hagedorn from the Denver Museum of Nature and Science, curator of geology at the Denver Museum. And he's going to talk to us all about fossils. And
thanks, Joel.
Yeah, and we left our last talk off talking about how much I like the Tapeats Sandstone And you said you were going to talk a little bit more about it here today. So in honor of that, I decided to go down here to Salt Creek, in the Tapeats Narrows of Salt Creek. So thought that was appropriate.
Excellent. What a
Yeah.
vision. And there's no better place to look at the Tapeats Sandstone than the Grand Canyon, right?
Exactly. Yeah.
That is
So,
one
yeah.
delicious looking outcrop behind you there. Someday let's hike down that, but maybe after the pandemic's over, okay?
That would be awesome. Yeah, that'd be awesome. So,
Super.
but yeah,
Well, thank
thanks
you
for,
so much, Joel. And thanks to everyone for tuning in. My name's James, and I'm looking forward to talking to you about some rocks and fossils today. And, one of the things that I often tell folks when I'm out in the field or leading a field trip or doing a tour is that, in reality, I'm just a detective in deep time. And, I'm tapping into that same curiosity that you have and that I have and that we all have. And that is, I'm curious about how do things work? How did they get that way? Why do they look like that? How will they be in the future? And as geologists, I'm kind of like a detective in time, deep time. Paleontologists are detectives in deep time too. And I'm kind of a hybrid one of those creatures. And so, but instead of DNA and a fingerprint or a footprint or a hair fiber at the crime scene, I look at rocks and fossils and minerals and chemistry and structures and things like that to figure out, what was the Earth like in the past and how did it change through time? And, fossils are one of the coolest clues to deep time and how our planet has been in the past and how it has changed over time. So are minerals. And so today I want to talk a little bit about fossils, which are, of course, awesome and super fun. They're basically dead critters from deep time. And I want to tell you a little bit about what some of the fossils are like in some of the lowest layers of the Grand Canyon in the Cambrian period from about 500 to 550 million years ago, maybe 540 or so. And some of the work we've been doing down there and that other geologists and paleontologists have been doing for years and why it's relevant to understanding how our planet operates. And I want to talk a little bit about the process that we go through when we're looking for fossils down there and what we actually use those fossils to do in partnership with other clues from these deep time crime scenes. And of course, part of any good detective at a crime scene is figuring out who did it, why they did it, how they did it, what was the murder weapon, and most importantly, when did it happen? Maybe not most importantly, pretty important. And to figure out the when, we need a clock. And so minerals, it turns out, are good clocks in deep time, and we'll kind of weave that into the story as well. Okay, so I'm going to share my screen with you, and then at the end of this talk, I'm hoping to connect with some questions. Maybe Joel and I can talk. Maybe you can send in some questions on the chat, and boy, it'll be a lot of fun. All right, so here we go.
All right.
There we go. And here we are. I'm down on the boat at the bottom of the Grand Canyon with Carl Karlstrom. We're rafting down the river, and we're looking up at the walls, and hopefully you can see my mouse, and we're looking at this big oozy, blobby looking thing right here. Oh my gosh, that's like a really bumpy surface. And one of the things that's unusual about this bumpy surface here, which we talked about just the other day, is that bumpy surface represents the erosion or the cutting down into these otherwise horizontally layered sedimentary rocks that were laid down by the sea layer by layer. And so it represents a gap in a moment in time. And just as that's a clue to kind of an ancient canyon or an ancient valley process, there are lots of other clues in these rocks. And so I want to walk through how we build a story from those clues, focusing in on fossils, and in this case, fossils from the Cambrian. So here's a view from right across the way. We're looking from just across the Bright Angel Trail, and there's one of these valleys here. This is Cheops Bay, and it's filled with these layered sedimentary rocks. You can see them kind of filling in this very shallow bowl-shaped depression. This is also known as Utah Flats. It's a very special area in the canyon that we've been tuned into for the last, 20 years or so with some of our colleagues in trying to figure out, well, what kinds of environments do these layers represent? And what kind of environments and changes are represented by some of the layers in the Tapeats, the Bright Angel, and the Moab formations that were behind Joel in his slide earlier and that are in the background of this slide right here. So in order to figure that out, we go out with a team of colleagues and students, and this is Aaron and Carol from Utah State, and we're measuring layer by layer these rocks, sort of describing their characteristics. Are they made out of sand particles? Are they made up of little shells that are glued together? Are they made out of mud that's gotten kind of squashed into what's known as a shale? Or are they a completely different kind of rock, like one that came out of a volcano or came through a crack or a fissure in the earth? And so we're measuring these layer by layer, we're taking notes, we're using Jacob stabs, and we're collecting samples along the way, because each of these layers is kind of like the pages of a book. And just as the story starts at the front of the book and it ends at the back, layer by layer, the layers of sedimentary rock in the earth do the same thing, from the oldest at the bottom to the youngest at the top. And so we take all this information down into a field notebook, making what's called a graphic log, or an illustration of how these rocks look layer by layer, and noting where we take samples to either date the rock or study their chemistry or study their environmental clues, or where we collect fossils and the like. And so this is just an example of one of those things. And so as we're going through these, we're also looking for ways to represent this that other geologists can understand. And so we might come back from the field and go to the lab and draft up this thing over here on the right here. This is called a graphic log. And the long axis, or the bottom to top axis, is the relative thickness of the rock in this case in meters. Each meter is about 1 yard. And then the different rock types have different symbols, little dots for sandstones, little dashes for mudstones, and so on and so forth. And I'm going to show you this diagram again and again in this talk, because it's kind of like the spine of because the spine of a book holds the pages together. And so in this case, this diagram is like a virtual spine for all the information that we want to integrate about the bottom layers of the Grand Canyon as we figure out what's happening in the Cambrian. So we go out in the field and we break apart lots of rocks and we look at them with our hand lens, and in between the layers of sand here are these little white blobs. These look like little shells. That's because they are little shells. These are not tiny little clams, but they're kind of things that look like clams called brachiopods, or lamp shells. And these are sea-living creatures that were alive in the world's oceans 500 and some million years ago. And in fact, there's cousins of theirs or descendants of theirs still live in the ocean today. And so when we find these in a rock, it tells us that this crime scene has fossils from the bottom of the ocean in it. Does that mean that that rock was deposited in the bottom of the ocean? What do you think? It might. It turns out that these fossils can be transported, just like a sand grain can get broken off a piece of granite at the top of Pikes Peak and then tumble downhill into a river, and then the river changes course and it dries up and it blows over here, and then something else grapples onto it and it moves downstream, and then it blows downstream, and then it goes into a river and then to an ocean. That sand grain has a journey from parent to source, or where it ends up in a rock, that may involve many different cycles and paths through different environments. And so fossils that are what are called body fossils, or things like these shells, can be transported from one place to another. In fact, here's an example of that right here. Here are a bunch of these clamshell-shaped fossils called brachiopods in cross-section, and in these, indeed, in this case, these have been tumbled together into a layer that sort of looks like a pile of cookies that's been tossed onto the counter, and everyone's kind of dug in there and pulled out some cookies but left a big pile of them behind. And so these layers have fossils in them that are from marine life, and so they suggest that if this rock wasn't deposited in a marine setting versus a terrestrial or land setting, that maybe it was very close to one. So to kind of figure this out further, we've got to look at more rocks. And so here's Karen from Boise looking at a rock, and she's peeled back the slab here, and there's nothing more exciting than peeling back a layer of rock and seeing something like this on it. I know, pretty cool, huh? These little blobs, they kind of look like little turd strings, don't they? Wait, am I allowed to say turds in talks? Or poop? Well, in fact, these things are sort of like that. These are the burrows of critters probing down into the mud, and these are little tunnel-like structures, kind of like submarine gophers, except in this case, these were probably more like submarine cockroaches, chewing into the muds at the bottom layers of the Grand Canyon back when it was a sea 500 million years ago. And so these fossils don't get transported, unlike those brachiopod shells I showed you before. And so these fossils, kind of like a footprint of a robber at a crime scene, let us know who was at that crime scene at that moment in time. And in this case, these are the burrows and feces and other movement paths. So in this case, this is some crawling tracks over here, and this is kind of a resting or a snoozing trace over here, and these are some trails of something kind of furrowing along in the mud like a little bulldozer. These let us know about the organisms that lived at that site at that moment in time, and at their activities that occurred there. And they haven't been moved. And so in fact, we do know that these rocks were mostly deposited, in this case, in shallow marine or shallow sea-like settings. And so that's really nice. That tells us a little bit about the crime scene and who its inhabitants were. And some of the adjacent areas in the field that we've been looking at actually have these huge cross-bedded or crisscross layers of rock that you can see under Zechariah here. And these crisscross layers of rock, which are underneath his feet, are actually formed at the bottom or at the toe of huge dune fields like the Sahara or the Gobi Desert or the Namib Desert. And what's cool is on the tips of some of these tracks, or some of these dunes, you can actually see tracks of creatures crawling up and down the dunes. And so in this case, here's a creature that wasn't living in the ocean or the sea, but was living adjacent to it, maybe in some coastal dunes, maybe crawling up the tracks like a beetle. I don't know if you've ever been camping on the beach at night, but I remember getting up to pee in the middle of the night once when I was a kid on the beach and looking around, and there are all these little creatures crawling around the sand, and well, it sure made me get back in my sleeping bag pretty quickly. Whew, zip it up. Anyways, so here's one of these creatures, but from 500 and some million years ago. How cool is that? These are the inhabitants of our crime scene. So we take this information together with information about the sedimentary rocks and their structures and their relationships with one another, and we try to come up with a picture of what the Earth looked like at a moment in time. And so in this case, this is an image from my friend Eben Rose of what the Grand Canyon setting might have looked like if you were in a time-traveling helicopter during the deposition of the Tapete Sandstone, and then later on the Bright Angel Shale and its friends. And so in this case, we've got some hills and mountains sticking up and some rivers draining in and around them, and they kind of intersect beaches and coastal dunes and then go off into the distance here into a shallow sea. And so we can put this information together and see how these environments and fossils change over time by putting them into our graphic log over here on the left, which represents those changing environments and changing fossils through time. But we still are kind of missing the clock part of it. And the other thing we're missing is, how does our crime scene, or how does the bottom of the rocks, the bottom of the Grand Canyon, relate to what's going on in the rest of the Earth at this moment in time? So to do that, we want to look for some fossils that are found all over the globe and that people have studied for just ages. And those are the cockroaches of the sea, known as trilobites. And so here's my friend John Foster cracking open a rock, splitting it, and he's hoping to get the bonanza, one of these squashed cockroach-looking critters known as trilobites that crawled around on the seafloor or burrowed in the mud hundreds of millions of years ago. And what's cool about these is that they break apart. And so trilobites are like many arthropods, like crabs and lobsters and shrimp, in that instead of growing and kind of keeping their body and adding onto it, what they do is they kind of grow for a period, and then they shed their hard skeleton or exoskeleton, climb out of it, and then grow a larger one, and so on. So they grow by molting. And what a cool discovery is it when you find your very first trilobite. Look at this grin. What an amazing, amazing find. This is Mike Moore from Boise with his first trilobite head. How cool is that? Can you see his grin? It's the size of the Grand Canyon. Someday you guys might find a really cool fossil too. Although you're not allowed to collect these in a national park unless you have a special scientific research permit. So in any case, here he is. He's found one of these. John found some. We've collected these layer by layer, and we put together what's called a range chart. And this is an example of this over here. So each of these kind of unpronounceable names at the bottom are the names of the types of fossils, like Homo sapien or Tyrannosaurus rex, or the trilobite equivalent of those names. And then the bars represent the thickness of rock through which we found these particular fossils. And this is an example of a really beautiful one here that Mark Webster found, and kudos to him for letting us borrow this image. And so we figured out how these trilobites appear and disappear throughout the sequence, but we need one more element to be able to relate these rocks to other places and to know how fast things changed. We need a clock. So I know Mark Schmitz and others and George Gerrels are going to talk a little bit about that, or maybe they have already. But here's Mike bagging and tagging some rocks here, which are going to go back to the lab. Those rocks are going to get crushed up. Here's a bunch of interns at the DMNS, many of which are from Colorado College or Brown, and they're crunching and munching these rocks and slicing and dicing them up. It's really fun to cut rocks, by the way. And they're breaking them apart, and then these rocks have all their elements separated out, including the heavy minerals and the lighter minerals and all the other stuff. And usually what you get left behind are a bunch of crystals. And so this is an example of some of these on the right. There are these very special crystals and rocks called zircons that are originally spewed out of volcanoes or that crystallize in hard underground volcanic rocks like granite. And they are very amazing because inside of them is basically an elemental clock that's been ticking since the moment it formed. And so we pick these out underneath the microscope and we put them in little hockey puck like mounts of really, really hard glue called epoxy or other material, and we might zap them with a laser to burn off some of that internal clock material, or we might put them into a mass spectrometer to like completely dissolve them out to let those elements out and measure their relationships to one another. And kind of like in a hourglass, that you know that when there's not very many sand grains at the top of the hourglass and there's a lot of sand grains at the bottom, that you're almost out of time, you can measure the same relationships of those components of these grains using these types of scientific instruments to come up with an age or a number of how old your rock is. And the idea is that you go layer by layer, and you do this at a variety of layers of rock to figure out, okay, that one's 510 million years old. That one's 509 million years old. Oh wait, that one's 412 million years. Whoa, there's a bunch of time in there that we don't know about. And so this process is called geochronology, and we pair it with the study of the fossils in layer by layer, which is called biostratigraphy. And our team has been revisiting doing this with some of the oldest sedimentary rocks that are horizontal at the bottom of the Grand Canyon. And this is some of our preliminary data. So this red number right here is an age that we very recently got out of the Tapete sandstone near the bottom of the Grand Canyon of 507,680,000 years, plus or minus 360,000 years. Hey, what's 360,000 years between friends, right? To a geologist, that's precise. To an anthropologist, that's terrible. In any case, so now we know that all these fossils down here that are in blue are slightly younger than that. And so the next step in the process is to date another rock above the fossils, within the fossils, to figure out how fast did these fossils appear? What do they tell us about the explosion of life that occurred in the Cambrian period? And perhaps lastly, how do they relate to all the trilobites that are found all over the planet? And so this is a really fun picture our friend Gerd Geier put together recently. And this is what's called a paleogeographic map. This is what the Earth looked like approximately from, if you were out on the moon, 500 and some million years ago. And it's been stretched out a little bit so you can see all the continents, but North America is over here on the right, and it's kind of turned on its side about 90 degrees. And the types of trilobites that we've been finding in the Grand Canyon are represented here by these little blue trilobite heads. And they've been found all over, and kind of cousins of theirs have been found on other continents, what's today Siberia, or Australia, or China, or Africa and South America. And we've just started the process of relating these trilobites and these ages to data from other continents to try to figure out what did that Cambrian crime scene look like. And you know, I want to give a shout out today. I don't know if you guys can see my t-shirt here, but this is a shirt with a big old trilobite in the front that my friends up at the Tate Museum up in Casper, Wyoming put together. So if you're ever up in Casper, Wyoming, get one of the coolest Cambrian t-shirts ever. Anyways, thanks for tuning in. Looking forward to your questions. Have an awesome day.
All right, awesome. Thanks very much for that. I would like to get up to Casper, Wyoming and get one of those shirts because that's a really cool shirt.
You want to see the back of the shirt?
Oh, well yeah.
Oh my gosh. So here we go. Not sure how this is going to work here. So the front says Spineless Wonders, and my friend Russell Hawes made this. Very cool. Oh. And the back, drum roll, please. Going to say, no, we're just going to have to find out.
Oh, the anticipation's killing me. Oh, nice.
Woo.
Nice. I definitely want one of those.
542. That's about the age of the bottom of the Cambrian, right when things got kicking and when the Cambrian explosion of life started. So in honor of a Cambrian talk, I thought I'd wear my Cambrian gear.
Absolutely, yeah. Now, I mean, the last thing I need is another T-shirt. I have a huge collection of them, but that one I make an exception for. And so, no, very cool. Yeah. Now, actually, the first national park I ever worked at was in Wyoming. It was a fossil site in Wyoming. So maybe you're familiar with it, Fossil Butte National Monument. Yeah.
Yeah. Did you work with Arvid?
I did work with Arvid.
Oh my God, Arvid is the best.
I know.
Crazy.
Arvid's amazing.
He's the best.
Yeah, and that was my very first position actually working with the National Park Service, was there at Fossil Butte National Monument. And if anyone likes fossils, it's an amazing place to visit. They don't get a lot of visitors.
If you guys like fossil fishing in particular, I mean,
yes.
Fossil Butte is known for what's called the Green River Formation, which is about a 50 million year old series of fossil lakes that have all these amazingly preserved fish and little leaves, and there are even birds, and sometimes there's reptiles, and it's a bunch of old fossil lakes, and they've got insects like you would not believe. And there are actually places outside of the park that you could go with your family. I've gone with our minivan and kids, and you're legally allowed to collect like on a private ranch. And if you find anything really crazy and unusual to science, you're supposed to give it to them, to give it to the museum or to the park, which they do. But you can go collect leaves and so forth. And anyways, the park's amazing. So what a great space. What a great place.
Yeah, awesome. And of course, that's much younger than what we're dealing with here at Grand Canyon. It's just a little bit younger. It's only about 50 million years old instead of like what you were talking about, 500 some.
And that deposit is exposed in Colorado, in northwestern Colorado, and in northeastern Utah. And what most people don't know is that we have a couple fossil-themed national parks here in Colorado, one of which is about 10 million years younger. It's called Florissant Fossil Beds, and it's, again, a series of lake deposits that are full with spectacular leaves and insects and all sorts of things. And so if you're ever in Colorado or Utah, check those out, everyone. Super cool, super fun.
Now, unfortunately, I haven't made it to Florissant yet. It's on my list. I think it's one of the only national park sites in Colorado I haven't actually gotten to. So that's high on my list of places I want to visit. So,
So what's your favorite? I got to know, Joel, what's your favorite fossil from the Tonto group? Do you have one?
Ooh, from the Tonto group. I don't know if I have a specific one. I mean, anything involving trilobites is always really cool. Not just the Bali fossils or the molts, like what you were talking about, but I mean the trace fossils, those tracks and resting traces that they leave. Those are really cool. Like down at Plateau Point. I remember I was down there for sunset one time out at Plateau Point, and I looked down at the peats beneath my feet, and there was just trilobite traces everywhere. There's really cool. There's really cool like walking traces. The ones that move along
are called. Here's how to remember. The ones that are moving along are called Cruziana.
Cruziana.
And the ones that are they're kind of heart shaped and plan view, and they're with the critter kind of bird in the mud. Those are called Rusophycus. Right.
Right. Yeah.
And ficus means plant, which is really weird because a long time ago people thought that all those tracks were fossil leaves or fossil algae. Really, the cruising traces of the trilobite or the burrowing traces of the trilobite. And then there's some other ones too, but those are the two. I know exactly what you're talking about. Those are so fun to find. Oh my gosh.
Now Cruziana I remembered, but
floor with your snorkel and your goggles, and you're like, okay, I'm in here, I'm breathing. Oh wait, a little trilobite just scurried along, and it's 520 million years ago.
Oh no, because trilobites are just so cool. And really useful, like what you were talking about for marking time, because there's so many different species of trilobites. And I like to talk sometimes, when we're here at the park, when we're talking about fossils, about index fossils, very special fossils that were only present on Earth for maybe a really short period of time. So when you find those, even if you're finding them on like different continents, you can match up rock layers. Yeah, kind of like.
And trilobites are. I always make the analogy that trilobites are kind of like insects today, right? If you want to study a bunch of environments and see change, you don't go measure elephants, right? Right? You go look at some kind of a moth, an invertebrate of some sort that is abundant and well distributed, and some of them, their distribution, and this is true for trilobites, some of their distribution is limited by time because they just went extinct quickly, that particular group, or they might be limited by environments. So for example, there are actually trilobites that hang out in the upper water column and swim, like agnostids and eodiscids. There are some trilobites that exclusively lived on the bottom, like in stinking black buds. There are some that would kind of foray into kind of the wave tidal zone of the beach, and there are other ones that live way out in the deep sea, right? And so there's a mix, and some of them do better in low oxygen settings, and some are better in well oxygenated settings. And so you're right, like they're really good proxies or clues to process, to environment, to time, and so fossils, I mean, along with minerals and structures and other things, are just great deep time clues to our planet. So
yeah, absolutely. And like what we were talking about before with how much you can learn from sedimentary rocks, and why I like them so much, is because I love, learning about, stuff up here on the surface of the earth, where we're living, and where other things are living or were living. And fossils, of course, they're huge. They're in helping us to interpret, what past environments were like and how they changed over time and what was living in them. I like to say, we look around on our planet today and we find different communities of living things living in different environments. And going back to that idea of the present being the key to the past, we can use what we see on Earth today to figure out maybe what Earth was like a really long time ago. Fossils are so helpful for that. So.
Yeah, I love it.
Now, and I wanted to be a paleontologist as a kid, actually. As I got older and my interest kind of broadened a little bit, I decided to study geology in general, but I have a special place in my heart for fossils. And I love studying them. I grew up going like basically every summer down to Washington, D.C. to the Smithsonian Natural History Museum.
Lucky you.
Geeking out over the dinosaurs, But I have to say, I've been to your museum there in Denver, and it's one of my favorite museums as well. It's been a few years,
Nice.
But
Yeah, I was really excited when I got a job here. My friend Kevin Peterson and I there's a scientific conference in Denver every year called the Geological Society of America meeting, every 3 years, it's in Denver. And we would come here and go to the conference, and we'd be stuck in some building downtown looking at slides or images and some dark hall in a hotel or the convention center. And every now and then we'd just be like, hey, Kevin, you wanna break out? Kevin would be like, yeah, James, let's get a cab. Of course, that's before Uber and Lyft, right? Or now it's like, let's walk there or take the bus or take Lyft. And we'd bust over to the Denver Museum of Nature and Science and just cruise around the exhibits or do something, or go to Dinosaur Ridge. And it's super fun.
Yeah.
The museums in many ways are a milestone in my life from place to place, for all the cities and countries I visited. So, and the Grand Canyon, of course, is a milestone for many people. My grandmother was the first person in our family to go to the Grand Canyon, and brought my mother there. My mother tells me the story of coming to the edge of the Grand Canyon, and their car did not have air conditioning, and this was a long time ago. And she went up to what I believe was the South Rim at the time, and she remembers that in the car they had little pans at the bottom, and she and her mom, now my grandmother who's now passed away, would sit at the North Rim, the South Rim, with their feet in pans of water to stay cool, because it was the hottest they'd ever been. Now, they were from Buffalo, New York, which is one of the colder parts of the country, so it was a little bit of a shock treatment, but they made this heroic pilgrimage, and my grandfather, Raymond Filsinger, said, you know what, we've got to go look at this. This is one of the 7 wonders of the world. And even if we don't hike into it, we want to take in the vista and appreciate what Mother Nature has done here. And I just feel so lucky that I get to work on it and be in it now and then.
So do I. Yeah, absolutely.
Yeah, and so for all of you viewers out there that haven't been to the Grand Canyon, it is truly spectacular. It is one of the 7 wonders of the world.
Yeah.
Enjoy it.
Yeah. And thanks for the intro to a little bit of geochronology as well. Of course, we're going to be getting into that a lot more deeply tomorrow. Be pretty much all about telling time tomorrow. So
good.
Yeah.
Well, hey, Joel, I've got construction at my neighbor's going on right now. So, and readers, hey, if you want to hear some saws going, that's what's happening. But I'm going to turn off my mic now. And thank you all for joining us. If you've got any questions, log in the chat. We're around, or look us up at the museum, Denver Museum of Nature and Science, or the National Park Service. So next time you're in the area, thanks so much.
All right. Thank you very much, Dr. James Hagedorn, everybody. Thank you very much again.
Well, hello again, everyone. Hopefully you're having a blast celebrating National Fossil Day with us here at Grand Canyon National Park. Now, we've had a couple of great talks already today about microfossils in some of the oldest sedimentary layers down there towards the bottom of the canyon, trace fossils made by things like worms and trilobites at the bottom of ancient seas. And our next talk is going to be focusing on what I think is one of the more fascinating types of fossils in Grand Canyon: footprints. Fossilized footprints or trackways left behind by ancient animals. And I think these are really, really super cool. And our next talk is extra special because it's focusing on a very specific trackway. It was fairly recently discovered, and the paper on it was just published by the next speaker. And it got a little media attention when it was published just a little while ago, so you may have heard something about that. But these are the oldest vertebrate footprints in Grand Canyon National Park yet discovered. They're in this rock right here, sitting along the Bright Angel Trail. It's a block of supai sitting right here along the trail, and there are actually two sets of footprints in this rock, as you'll soon learn in the next talk. But like I said, oldest vertebrate footprints yet discovered in Grand Canyon. It's going to be a fascinating talk. You're not going to want to miss it, so stick around. Thanks for joining us.
All right, well, hello, everybody, and welcome to another talk in our series of presentations, of talks for Grand Canyon Geology Festival 2020, Earth Science Week 2020, and one of our talks, of course, here today for National Fossil Day 2020, and we're here with Dr. Stephen Rowland, and discussing some of the oldest trackways, the oldest fossil footprints found within Grand Canyon. Dr. Rowland, thank you very much for being here with us today.
Thank you, Joel. My pleasure.
Yeah. All right. So let's, if you're ready to go, let's jump right into your talk here today.
Good evening. This evening I'm going to tell you about some very cool fossil footprints that I've been working on with my colleagues in the Grand Canyon. And if you know anything about trackways in the Grand Canyon, you probably know about the Coconino Sandstone. Here's a view from the Bright Angel Trail that shows the Upper Paleozoic part of the geologic section. The Coconino Sandstone is famous for its fossil footprints. They're quite abundant. It's a wind-blown sand dune deposits. But the tracks that I'm talking about this evening are much lower in the section. They're down in the Supai group. The Supai group consists of four formations: the Esplanade, the Weskagami, the Manicacha, and the Watahamaji. The tracks that we're talking about this evening are in the Manicacha formation, which is a bit surprising because the Manicacha has never produced tracks before. Tracks have been found in the Weskagami formation back in the 1920s, but nobody has ever seen tracks in the Manicacha formation. So it was a surprise when these tracks appeared. The tracks occur adjacent to the Bright Angel Trail in the boulder on the right of the center of the view here. These tracks were first alerted to me by my friend Alan Krill, who's a professor of geology in Norway, and he was hiking on the trail with his Norwegian students a few years ago and noticed these tracks. They're very conspicuous, and anybody who looks at them would immediately know that they're fossil footprints. So Alan saw those, and he knew I was interested in fossil trackways, so he alerted me to them, and I went and studied them and have learned quite a lot. There's actually two sets of trackways on this boulder, and I'll talk briefly about them. Trackway 1 is the most conspicuous set of tracks, but there's another trackway that you wouldn't notice unless you looked really closely, and that's what I'm calling Trackway 2, which consists exclusively of claw prints of an animal that walked on that same surface. I'm going to turn this view, rotate it clockwise, so that now all the tracks are headed up toward the top of the slide. But notice that the main trackway number 1, the tracks are pointing upward, straight to the top, but the whole trackway slants about 40 degrees to the right. So this animal was walking sideways, so to speak, as it moved along, and I'm going to talk about our interpretation of that. Here's a photograph of the tracks. This is trackway 1. The LM stands for left front, or left manual, left manus, and then right front, left rear, and right rear. And our interpretation is that consistently the left front print is on the left end of each of the rows of 4 tracks, and the right rear print is always on the right end, and so on. So these tracks maintain their position within this trackway in our interpretation. Some people have looked at this trackway and interpreted it to be the result of 2 separate animals, either walking side by side or in close succession, but that would require precision stepping where they didn't step on each other's footprints at all, and my colleagues and I have rejected that interpretation. I'm only going to have time to talk about trackway 1 this evening. Trackway 2 is this other trackway, which has its own interesting story. We recently published a paper just a few weeks ago in the journal PLOS One, in which we describe both of those trackways. PLOS One is an online open access journal that anybody can download, so you're welcome to read all the details in that journal if you'd like. I'd also be happy to send anybody a PDF of that article if they want to contact me by email at UNLV. So the questions that we're addressing about these trackways this evening are as follows. First of all, we want to know, of course, how old are they? We want to know what kind of animals made them. What's up with this weird 40-degree sideways gait? What was the depositional environment that these animals were walking in? And how significant are these tracks locally and globally? And finally, I'll end with a brief comment about management issues. So first, let's address the question of how old are they? The geology of the Bright Angel Trail has been mapped in great detail, and that's been very helpful. And this diagram is what's called a time rock diagram that shows how rocks are distributed through time in the Carboniferous and Permian periods. We'll focus just on the Carboniferous, and in fact the Permian epoch of the Pennsylvanian epoch of the Carboniferous period. And you see the Manacacha Formation is right here in the lower part of the view. The pink color shows wind-blown deposits, or what we call eolianites. So you can see eolianites occur in various formations in the Grand Canyon and also in the Manacacha. But in addition to the eolianites, or wind-blown deposits, there are marine deposits in the Manacacha, which contain fossils that are age diagnostic. So it's the fossils in the marine rocks that have helped geologists determine how old the Manacacha Formation is, and it turns out that it belongs to the Atokan stage of the North American geologic timescale. The Atokan stage is 319 to 312 million years ago. And also you can see in this diagram, the blue band represents the Manacacha Formation. That red line within the Manacacha Formation is the horizon in which these fossils occur. So they occur about 3/4 of the way from the bottom to the top. The Manacacha Formation itself is 316 to 312 million years old, and so 3/4 of the way up would make it about 313 plus or minus 500,000 years. So that's how old we think these footprints are, 313 million years old. Notice that 313 million years is just slightly, a million years or perhaps even less, younger than the oldest undisputed amniote skeletal fossils in the world, which occur in Joggins, Nova Scotia. Skeletal fossils refers to bones and teeth, whereas what we're talking about today are trace fossils, such as footprints. I'll talk about amniotes in just a minute. So that's how old we think these fossils are, about 313 million years. And that's mid-Pennsylvanian epoch within the Carboniferous period. So the next question is, what kinds of animals made these tracks? And we go back to this diagram, and often when we're trying to interpret what sorts of animals made any kinds of tracks, the first question we ask is what animals were available at the time that might be candidate track makers. And in this case, there's a bunch, but they have a common feature in that all of the candidate track makers are amniotes. Amniotes are animals that laid a shelled egg. A shelled egg such as it could be a kind of a leathery egg such as a turtle egg, or it could be a hard-shelled egg like a bird egg. But the presence of the amniotic egg, the egg that with the shell on it, allowed these animals to live outside of an aquatic environment. Their ancestors were amphibians, which such as salamanders and frogs and toads, which of course have to lay their eggs in water. So amphibians are tied to a watery habitat so that their eggs can be deposited in water and the animals go through a tadpole phase. Amniotes don't do that. They can live in an area without water, such as a desert, for example, because the eggs have a shell on them which protects them from drying out. So the earliest amniotes come in, as you can see, at about 325 million years ago or so, just a few million years prior to our track maker. We cannot distinguish in our tracks between reptiles and synapsids. Synapsids, as you can see on the left, are kind of the sister group of reptiles, and they include animals such as pelycosaurs and therapsids, and our own ancestors, mammals, are synapsids. So this is the line toward the left. The synapsid branch is the branch that goes to humans and mammals in general, whereas the reptile branch goes off to the right. At this stage in the evolution of these amniotes, we can't distinguish the footprints of synapsids from reptiles. So we can say that these are amniotes, but we don't know whether they're early reptiles or early synapsids. So what's up with this weird 40-degree sideways gait that we see in trackway number 1? Well, our interpretation is that the way this track was made was as shown in this little diagram I'm going to show here. If you imagine the animal standing still with 4 legs underneath him, the first step is a right rear leg moving up, forward, and to the right, followed by the right front, then the left rear and the left front. Right rear, right front, left rear, left front. Right rear, right front, left rear, left front. That's called a lateral sequence gait. Lateral sequence because the legs on one side of the animal are moving in succession, followed by the legs on the other side. If you have a cat or a dog at home, and you watch that animal move slowly around the house, you'll see that animal using exactly this gait, not necessarily moving to the right, of course, but they use a lateral sequence gait. It's a very, very common gait when tetrapods move slowly. It's not the gait, though, that we inherited from our fish ancestors. That would be a diagonal sequence gait where the right front is followed by the left rear and the left front by the right rear, and so on, or they move in succession or synchronously. But a lateral sequence gait is a very interesting gait that looks like this. And if you continue this lateral sequence gait, as you see in this little pseudo animation, you see that we can develop, if the animal is moving to the right, we can develop a pattern of tracks identical to the trackway number 1 pattern. So this pattern of tracks, in our view, is most compatible with a locomotion model in which a single animal, not two animals, was using a lateral sequence gait, and each step was offset about 8 centimeters to the right. This is the oldest known example of this type of gait. So we know now that tetrapods were doing a lateral sequence gait as early as 313 million years ago. So what was the depositional environment in which these animals were walking? We need to look at the sediments to figure that out. And there's basically 2 kinds of deposits. This is on the left is a stratigraphic column and a weathering profile of that particular section of the Bright Angel Trail where the Manacacha Formation is exposed. And you can see the yellow pattern is cross-bedded sandstone. In this case, it's wind-blown cross-bedded sandstone, and the purple color is mudstone deposited by standing water. So it's a combination of these two types of sediments. And here are some sedimentary structures that are exposed in the wall of the Manacacha Formation along the Bright Angel Trail, close to where the trackways occur. And these are all indicative of wind-blown sediment. So there's no question that this was an aeolian setting or a wind-blown sand dune type setting. Here is a petrographic thin section where we'll take a piece of rock and slice it down to the thickness of a piece of paper and look at it through polarized light and plain light. All these grains, all the light colored grains in the bottom diagram and even the black and gray and white grains in the upper diagram are quartz. So these are rounded quartz grains, very, very typical of wind-blown sand dune deposits. So there's no question that the depositional environment was a coastal dune field, and we have an artist reconstruction by Emily Waldman showing an early amniote walking diagonally across a dune face. And our interpretation is that this animal was walking diagonally to decrease the gradient of the ascent. So when we go hiking in the mountains, we don't go straight up the mountain. The trail switches back and forth. We do switchbacks. And so this is what this animal was doing, was a switchback. He was walking diagonally, presumably to make it a little bit easier for it to go up the slope. We can look at the paleogeographic distribution of rocks and ocean and different environments back in the Atokan period of the Pennsylvanian epoch, the Atokan stage rather. This is a map courtesy of Ron Blakey. And if we can look at close up, we can see that the Bright Angel Trail track site and the Manacacha Formation in general is represented by a dune field and tidal and estuarine environments along the eastern edge of what's called the Grand Canyon embayment. So that Grand Canyon embayment has nothing to do with the modern Grand Canyon. It's just a marine embayment that is close to where the Grand Canyon is now. And that explains or further justifies the environment where we have wind-blown sand that is periodically interrupted by mud that was deposited perhaps either by high tides or maybe a flood waters coming down a drainage system in the same area. So how significant are these tracks locally and globally? We argue that these are extremely significant tracks. They're the oldest vertebrate tracks in the Grand Canyon, which is saying quite a lot. They are among the oldest amniote tracks on Earth. They're the oldest example of amniotes living in wind-blown sand dunes. Prior to the discovery and study of these tracks, it was not known when amniotes first started inhabiting sandy environments. The previously known earliest example was about 8 million years younger than this in northeastern Utah in the Weber sandstone. So this is the oldest example of wind-blown sand dune dwelling amniotes. And finally, it's the oldest example of a lateral sequence gait, this very distinctive gait that is so common these days in modern tetrapods. Just to end with a brief comment on management issues, this trackway is obviously very vulnerable to vandalism. It lies right next to the most popular trail in the Grand Canyon, one of the most popular trails in the world. Hundreds of hikers pass by it every day. And it's a valuable resource that needs to be protected, in addition to the fact that it's weathering away. So eventually, after a few decades, this trackway will be much less distinct than it is today. And so we're in discussion with the National Park Service with the idea that this trackway should be protected. So we're hopeful that it will be helicoptered out of there and eventually put on display in the Geology Museum at the South Rim. So thank you very much. It's been a pleasure.
All right. Well, thank you very much, Dr. Rowland. Yeah. So no, really, yeah. really exciting stuff. I've had an interest in paleontology since I was a little kid, and I wanted to be a paleontologist when I was a little kid. I ended up deciding to kind of broaden my field of study and go straight geology. But I do really love paleontology, and I was actually a TA for 2 years for undergraduate paleontology as a grad student. So I love this stuff. Like, I really love this stuff.
Thank you.
And
Cool.
yeah, and no, I definitely hope. I kind of hope that we do as an agency, as the Park Service does, take your advice and move that block. I mean, it's not in situ anymore anyway.
Right.
To
There are
help
actually,
preserve it.
I didn't get into it in my talk, but there's actually two blocks. The opposite, the other side, the top of that block that popped off when that rock fell, it broke in two right along that bedding plane, and the other side, the counterpart block is a little bit ways down the hill that shows natural cast, the little bumps that fit into those impressions. So I think it would be a wonderful exhibit to have a kind of an open book type exhibit with the main rock that's now on the side of the trail as the lower block oriented horizontally, and the other one oriented vertically or nearly so you can actually see how they fit together with an interpretive story about it. I think it'd be a good
Absolutely, yeah. No, that would be a really great display. Actually, the first national park site that I ever worked at was a place in Wyoming called Fossil Butte National Monument.
I've never been there. Yeah, I know of it. I've never been there.
It's a fascinating place if you like fossils. Yeah, they have some great exhibits like that, where they have the mold and the cast, paired and then split like this. They've got a giant, like 6-foot palm frond that's split like that in the visitor center there. Yeah. One question I did want to ask was as somebody who's in part of my thesis research doing some geochronology work about the dating methods used to pin down so precisely that date of 313 plus or minus 500,000 years.
Right. So, it all comes from the work of other people, Ron Blakey mostly, and some other folks, but mostly Ron Blakey. And he published several papers on the Supai group and the age and so on. So the dating comes from marine fossils, I think probably mostly fusullinid forams, which occur, of course, in marine facies within the Manicachaw. And forams, as you know, evolved very quickly and are quite precisely dated to death by the oil industry paleontologists. So those dates of 312 to 316 for the Manicachaw come from dates that were derived by Ron Blakey and others because of the marine facies. And so because the trackway horizon occurs 3/4 of the way and up from the base, and assuming, and this is a fairly big assumption. I had quite a discussion with the reviewers of our paper about this dating issue, and it was good. I mean, it was appropriate for them to question that. And the original date I gave was 313 without any error bars. And what the reviewers said was you can't assume a constant rate of deposition. And they're right. They're absolutely right. And so what we had to do for our paper is get deeper into the literature and talk about why we think that the rate of deposition was at least fairly constant. And that involved discussion of marine rocks out in Nevada and the Birch Spring Formation, which have been studied in quite detail by folks. And so what we did is made the case that yes, we have some basis to assume a relatively constant rate of deposition, and because the tracks are 3/4 of the way from the bottom to the top, and the formation is 312 to 316, that 313 is plus or minus 500,000 is a reasonable best guess estimate of the age. So that's the basis of that.
All right. Oh, awesome. Yeah. And of course, those watching probably remember or who were watching the first day of the Geology Festival. Ron Blakey gave a talk on the Paleozoic rocks of Grand Canyon, changing landscapes over time.
Yeah.
All right, well, we've heard about some amazing fossils here in Grand Canyon. Microfossils down there near the very bottom of the canyon, trace fossils, that amazing trackway that was just recently discovered along the Bright Angel Trail. And our final talk here for today, for National Fossil Day, is going to focus on ongoing research in paleontology here at Grand Canyon by the National Park Service. Now, I'm standing here at a fossil exhibit on the South Kaibab Trail at a place called Cedar Ridge. Now, this fossil exhibit is actually really historic. It was established in 1937 by one of the first park ranger naturalists here at Grand Canyon, Edwin McKee, who also happened to be a geologist and, let's be honest, a little bit of an idol of mine. Now, he actually got the job in 1929. Unfortunately, the first park ranger naturalist, Glenn Sturdivant, passed away on a river trip. Edwin McKee went on to become one of the most iconic park rangers here at Grand Canyon, an amazing geologist and paleontologist who did a lot of incredible scientific work during his time here at the park. And this fossil exhibit that he conceived and installed was actually to honor another paleontologist named David White. So there is a long tradition of paleontology here at Grand Canyon, which Edwin McKee helped establish and which the Park Service today continues. And that's going to be the focus of our next talk by our park paleontologist on some of the most recent discoveries here at Grand Canyon by the National Park Service in the Paleozoic rocks that you can see behind me here. So enjoy.
All right, well, hello everybody, and welcome to our final talk here for today. It's National Fossil Day here in the middle of the week, or Earth Science Week. So, of course, here for our talks for Grand Canyon Geology Festival, GeoFest 2020, we are focusing all on fossils and paleontology here today. So we've heard a number of talks about some really cool fossils in Grand Canyon, some of the oldest fossils in the canyon in rocks way down there towards the bottom, and a few talks about some awesome trace fossils and trackways. Now here to talk to us about a lot of other kinds of fossils that are found in the canyon is our park paleontologist, Ann Miller. And she got her Bachelor of Science in Geology at Oklahoma State University in 2013. And after that, worked for a while for the American Geosciences Institute, the Student Conservation Association, and got interested in paleontology as a research assistant for Glen Canyon National Recreation Area. And working on dinosaur trackways up there sparked her interest in trace fossils. Started working as a paleontology intern at Grand Canyon National Park in 2016. Since then, she's completed her master's degree at Northern Arizona University, just down the road in Flagstaff, working on trace fossils in the Bright Angel Shale, and is continuing to work for the park as a contracting paleontologist. So thank you very much for being with us here today.
Thank you.
Absolutely. Appreciate you joining in and talking to us about what's going on in the world of paleontology here at Grand Canyon National Park.
It's my pleasure.
All right. Well, if you're ready to jump into it, let's talk about some fossils.
All right. Just going to share my screen here. Well, thank you so much, Joel. Today, I'm going to talk about the Paleozoic fossils of Grand Canyon, so mostly showing really cool pictures of fossils. And of course, we're going to start right into the Tapeats Sandstone. I should note here that the 60 Mile Formation has been recently part of the Paleozoic. It's been recently dated. We just haven't surveyed it yet. So not really sure what fossils are in there or what fossils are yet to be discovered. But as far as the Tapeats Formation, we have mostly trace fossils that we find here. And here's an example of a really common trace fossil called Paleophycus. And it's basically a network of burrows, probably made by some sort of worm, maybe a priapulid or something like that. And this is looking up at an overhang, and these worm burrows just covered this entire strata. And then down here, this is a little bit more rare trace fossil comparatively, called Rhizophycus, and this is a trilobite resting trace. And you can see some of the V-shaped striations in here represent the marks of the trilobite sort of digging in the sediment, probably resting here during some sort of storm or something like that. Now jumping straight into the Bright Angel Shale, since this has the majority of Cambrian fossils reported from Grand Canyon, we're at about 505 million years ago. And here's some iconic trilobites that we find in the Bright Angel, Glossopleura, Amaniella, and these are housed in the Smithsonian Institute. And obviously these ones are a little bit smaller. If you could see the scale bar down here is in centimeters. And here's some more of the body fossil record of the Bright Angel Shale. Keep in mind that these are all pretty rare. It's not very common that you'll find these. You might find brachiopods a little bit more commonly, but we have seen things like hyolithids, lingulid brachiopods, obviously, bradorids, which are arthropods, but they've been compared to ostracods, and you can see they're quite small looking at this 1 centimeter scale bar here. And so is this hyolithid. This is 1 centimeter. And here's a few other trilobites that you might find in the Bright Angel Shale. All right, so body fossils are still pretty rare in the Bright Angel Shale, but trace fossils are so common. Any outcrop you go to, you're almost 100% likely to find some sort of trace fossil, most likely worm burrows. And so I had to show Paleophycus, which again is probably at least the most common trace fossil that I've found in the Bright Angel. But we also have Rhizophycus again. You can see the striation marks. And then we have Cruziana. This is actually a really nice specimen of Cruziana. And it's very similar to Rhizophycus in that it was most likely made by a trilobite. But in this case, the trilobite is moving along the surface and sort of creating a furrow, likely as it's feeding. So it's moving along and feeding the sediment. And here's a few trace fossils that I found during my master's degree, actually. This one in the left corner is Bergeria. And this is often found within the bedding plains. And it's kind of this plug-shaped burrow. And it was thought to have potentially been made by some sort of cnidarian, likely a sea pen, that had sort of a rounded base where it could anchor itself into the sediment. And when it died, it then disintegrated and sediment would fill in the burrow, creating Bergeria. And then over here, we have another worm burrow that, as far as my master's work, this was the second most common trace fossil that I found, called Tychicnus. And these worm burrows, instead of Paleophycus, which is usually found horizontally oriented, either below a bed or in the top of a bed, Tychicnus is often found within the bedding plains. And it's distinguished by this sediment lamination on the side called spritin. And you could see that over here as well in this U-shaped burrow called Diplocriterion. You can see a little bit of that sediment laminae between the limbs of the burrow. And this spritin indicates that the creature that made these burrows were shifting in the sediment vertically in order to maintain its distance with the sediment-water interface, which makes sense if you think of like a filter feeder. If there was a filter feeder in this U burrow, it's going to want to stay near the surface. Whether there's deposition occurring or erosion, it's going to be shifting. And in this case, you can tell that it shifted downwards. So obviously we had some sediment deposited on top. And then over here, we have Trypicnus pedum, which is a feeding trace. And this one is interesting because it's actually used to determine the Ediacaran-Cambrian boundary. And in this case, obviously, this was found in the Middle Cambrian. so it can't be used for any sort of dating. But it is one of the first trace fossils that appear in the trace fossil record. Now this trace fossil is very interesting. It's called Monomorphicnus linnaeus, and its variation is giganticus, meaning that this is a very large form of this trace fossil. And it's essentially just these parallel striation marks on the sediment. In this case, I'm looking up under an overhang. So this is in cast. And yeah, it's quite large. Since they're all parallel, it was likely that it was made all at once, all of these striations. And so there weren't a whole lot of options. Since this is so large, there aren't very many large trilobites in the Bright Angel Shale, or arthropods for that matter, as far as the body fossil record. And so the best option that I could come up with was that this may have been made by a large predator called Anomalocaris, where it may have used its appendages to swoop down as it was swimming through the ocean, to swoop down near the surface and pick up its prey, whether it be a trilobite or a priapulid worm. And in this case, I used A. canadensis from the Burgess Shale, which is about the same time period as the Bright Angel Shale. And there's these spikes that come off the appendages, and the spacing measures pretty closely to the spacing here. And so it's actually quite likely that this creature, you can imagine this ferocious looking creature, very big compared to most other arthropods, you know, came in and created these striations as it was preying on animals or trilobites or anything. So it's just really interesting because we don't have any body fossil evidence of this creature ever existing as a Bright Angel Shale, but we have this really interesting trace fossil that tells us, hey, this creature might have existed here. Now moving on to the Muav limestone, we're still in the Cambrian, and I don't have a whole lot of fossils to show you from the Muav. A lot of it is highly bioturbated, so there are trace fossils. They're just a little bit harder to identify. But here are just some of the trilobites that we might find. And again, these are housed in the Smithsonian Institute. Now we're going to jump right into yet another ocean. We're in the Redwall limestone, so we're about 350 million years old. And here's just an array of some of the fossils we find. We find crinoids, crinoid stems and ossicles, gastropods, nautiloids. This is an especially large nautiloid. And then we find also fish teeth like this Helotus tooth here. And again, this is a 1 centimeter scale. And then we also find really beautiful coral impressions. This one was obviously mineralized and filled in. And now I'm going to jump right into the next depositional environment. And this includes most of the Supai group as well as the Hermit Formation. And what we're getting into is kind of a broad coastal plain. It was somewhat arid, but we also get rivers and swamps, and we get tons of plants, all kinds of creatures like insects and different synapsid reptiles and things like that. This is all existing in this depositional environment. And some of you might actually recognize this trackway here. This is the famous Rowland trackway, where he just put out a paper, Rowland et al. 2020, and so far it's the oldest vertebrate trackway reported from Grand Canyon, and he identified it as Chilicnus-like trackway. And again, this is from the Supai group Manacacha Formation, which is one of the lower formations in the group, and he estimated about 313 million years old. So that's an old trackway. But we do find other interesting trackways in the Supai group. This one's a little bit younger, likely found from the Esplanade sandstone. And you can see the footprints are much bigger, and it's in situ, so it was likely walking uphill. And so that is just a really cool one from the Supai group. But I'm going to jump right into the Hermit Formation, because we actually find a lot of fossils in the Hermit. And so we're about 285 to 280 million years old. And we find trace fossils like these burrows called Scoiana gracilis. These are feeding burrows. And then we also find really cool things like this dragonfly wing. And this scale over here is just 1 centimeter. So this was a very large dragonfly. And these specimens here, these are fossil plants. These were found just this year. This beautiful, large seed fern plant was found just this year as well. And it's one of our favorites because it's so large and it's so well preserved. And we think it's Supaia. And then this one over here is called Walkia. And this is a conifer plant. And again, we just found this one this year as well. And here's a few more plants. There's this horsetail, Sphenophyllum. And this was also found this year. What's cool about this one, though, is that the stem in the middle was found going straight through the slab, and you could see the stem on the other side of the slab. And so that indicates that this plant was buried in situ. So while it was standing there, it got buried, which is really interesting. And then, of course, we have neat trackways. You could see the toes or claws are kind of facing each other here. And this is a tetrapod trackway, kind of typical of what we find in the Hermit Formation, called Amphiseropus. And this was also documented in a recent paper. And now jumping into the Coconino sandstone, we're getting into about 280 million years ago. I show a picture of the Namib Desert because I think it's a great modern analog of what the Coconino sandstone looked like. So we're kind of near a coast, but we have this huge sand dune field. And surprisingly, there were a lot of critters walking all over. We have all kinds of trackways, likely made by a synapsid, some sort of early reptile. You can see in some of these, the claws are very well preserved. And then even these ones here are quite large. And a lot of these were found just within the past 2 years. So pretty neat. And we're finding more and more every time we go into the field. And of course, this one is sort of the iconic trackway of Grand Canyon called Ichneotherium spherodactylum. And it was identified this way mainly because of its rounded toes. And it was possibly made by an amphibian-like reptile called a diadectomorph, which kind of looked like these guys. And again, this is reported in a recent paper by Francis Cheney et al., 2019. Also note the scorpion down here in the corner. And that's foreshadowing for these trackways, which are invertebrate tracks, most likely made by different arthropods like millipedes, isopods, spiders, and scorpions. And you can tell that they actually had to be pretty big to have created these tracks. All right, now getting into another ocean. I'm going to end it with the Kaibab Formation. And again, this is sort of what the depositional environment looked like. Here's some of the invertebrates that we find. Bryozoans, sponges, rugose coral, and crinoids, of course. And then we find these brachiopods. These are actually now called Panicularis bassi. And these, again, housed in the Smithsonian. We also find brachiopods called Derbia regularis. But also there are trace fossils in the Kaibab limestone, such as this huge burrow network, which spans laterally quite a ways. And I identified this as the Thylacinnoides, which was most likely made by crustaceans. And then here's just an interesting fossil called Conulera caibabensis. And it's not very well known, but it was thought to have been made by a conularid, which is a cnidarian that kind of looked similar to this rendition here. And then, of course, we have vertebrate material coming out of the Kaibab limestone as well. We have shark teeth. We have this phyllodont tooth plate. You can see the individual teeth in here. This tooth, Gilcomanius, is kind of the iconic look of what you might find as a shark tooth, but it's very, very small. These are just millimeter scales here. But probably the most significant was this Megatenopetallus kaibabinus tooth. And it's significant because it was a tooth of a very large shark called the Kaibabvenator. And here's another rendition of the tooth plate. So this is kind of more what it looks like. Again, this is a centimeter scale. So it was a very large tooth. And this Kaibabvenator was just about the same size as the great white sharks that we find today. So ending with that, I just wanted to remind everyone that we do work under the Paleontological Resources Preservation Act of 2009. And so we are supposed to protect paleontological resources, inventory them, and use them for scientific and educational use, make sure the public is aware. So that's part of what this presentation is about, is to let you all know what kind of fossils we're finding. And if you wanted to do any research on these fossils, definitely let us know, because there is just a plethora of opportunities for research, especially since we're finding fossils every time we're in the field, and some of them have been spectacular. So just to let you all know, and then of course, if you find a fossil and you don't have a research permit, please leave the fossil there. And if you can, take a picture with GPS coordinates and then send that over to me. And that'll really help me out. I'll have that information. I'll be able to eventually go into the field, check it out, maybe identify it, and include it in our inventory. And so that would be really helpful if you could do that for us. And then one more thing I just wanted to add was that we just created a new Grand Canyon Paleontology social media page. So we have a Facebook and we have an Instagram. Just keep in mind that this is non-NPS affiliated. So this is mainly for paleo enthusiasts. But we do post really cool information about science, about fossils, and a lot of times we will post pictures of fossils that we've found recently. So really cool stuff if you are interested in following. And with that said, does anyone have any questions? And yes, these are all letters that I found in trace fossils. So enjoy.
All right. Awesome. Well, thanks so much for that look into the world of paleontology here at Grand Canyon. I appreciate the ending a lot. You know, those I always make sure to tell folks, you know, when we're looking at fossils here at the park, that they have to stay there. You can't take them out of the park. And of course, even if you keep them technically in the park, if you move them from one place to another, then that destroys a lot of the important information about them. So super important to just to look at them but not actually move them, or, you know, of course, not take them out of the park. So yeah.
Definitely.
Yeah. And of course, I got like, you know, 6 million some people in a normal year coming to the park every year. And I always like to tell people, I mean, if everybody took home one fossil with them, we probably wouldn't have many left for people to come and see. So
very good point.
But yeah, actually, I didn't realize, I didn't know about that, the Grand Canyon Paleontology Facebook page until just recently. I found it because a friend of mine shared something from it, and I was like, wait, what? What's this page? So no, I started following it myself recently.
Oh, great. Yeah, we just started it. So we're really excited to get that following. And we've started to get a really good following so far. So it's looking pretty good. Yeah.
Yeah, because I mean, a lot of people are interested. I get a lot of questions about, it's like, well, what work is going on? Like, what research is currently happening in the park? What are you looking at? So that's probably a great way to keep hopefully up to date on that.
Yeah, and feel free to tell them about our social media page, because we will update on some of that recent work. Especially that we're doing internally. So and that stuff's not very public, so it's a great opportunity to let the public know what's going on, that, hey, we have fossils and they're spectacular. So
cool. Yeah. And one thing I love about paleontology is how it helps us reconstruct ancient ecosystems. I mean, we can learn a lot by looking at geology. It's just, kind of the types of sediment that we're accumulating in a certain location, and the structures that we find in that sediment. But fossils are often such a critical part of trying to piece together, like, well, what was this part of the world like? How did it change over time? And I love how you presented it, in a chronological order, moving up through, we always like to talk about the layers in the canyon, like pages of a book or chapters of a book, each one telling its own story. And as you move up through, you're actually watching like how this part of the world changed over time, how the environment shifted from one to another. And also, I mean, even you're kind of getting at a little bit the evolution of life on Earth as well.
Yeah, pretty much. That's what's so amazing about the Grand Canyon is it's very rare to have such an amazing stratigraphic section showing almost the entire Paleozoic. Obviously, there's missing time in there, but
right.
It's such a great way to see how life changed and how the deposition and the environments changed over time. So yeah, I completely agree. Grand Canyon's very unique for that.
One thing I wanted to ask you about a little bit is those amazing trackways that we find in the Coconino Sandstone, which is actually an example of that right behind me here. This is a slab along the Trail of Time, right up here along the rim of the canyon. So the Coconino is not too far down below the rim, just a few hundred feet. So it's not too hard to hike down there and maybe see some of these trackways, in place. But for folks up here walking along the rim, this one actually got put right there on the Trail of Time so people can see these. But we always compare that environment to the Sahara in Africa today. Wind-blown sand dunes, along the coast. And a lot of people ask me, it's like, well, how did these footprints get preserved in an environment of blowing sand? And I wanted to get your opinion. My answer is always is that since it was a coastal dune field, something like a phenomenon that we see today, oftentimes in environments like that, is like fog banks forming out in the ocean, moving inland, coating the upper layers of sand with moisture so that they're wet, and then creatures crawling across them, those footprints get preserved and then very quickly covered up by blowing sand. Is that a good explanation, or?
Yeah, actually,
add anything to that?
I think that's a great explanation, because that's kind of one of the harder parts about the Coconino to be able to understand, especially with some of these tracks that are so well preserved. I often find myself wondering, how was this preserved this way? And really, it does have to do with the amount of moisture. So I tend to think that there probably was some sort of fog coming in, especially if a lot of these creatures' trackways are next to each other. So maybe, these animals were all going to one place to obtain this moisture or something. And the interesting part is that it couldn't be, you can't have too much moisture, but you can't have too little. So really it had to be the precise amount of moisture. And you can see some trackways are a little bit better preserved. So there might have been a little bit more moisture. Maybe the sand was maybe a little crusty. And so they were able to have really nice footprints in there. Other trackways are a little bit more messed up, and you can tell maybe the sand was a little drier. Sometimes there's a tail drag, and so you can see their footprints are a little messed up, but you can see this really nice tail drag in the middle.
Right.
And so, yeah, and if you think about it, if you're walking along the beach, you might see a little bit of your footprints, but as soon as that water covers it up, it wipes away the footprint. So we know that there couldn't have been that much water in order for it to be preserved. And because it was windblown sand, after those tracks, had some of that moisture, it was probably buried very quickly after that with that windblown sand. And those are the factors to get a really nicely preserved trackway.
Yeah.
You're on track.
And one other thing that you said that reminded me of things that I talk about a lot to visitors at the park is how rare some of these fossils are. Yes, Grand Canyon, I mean, is chock full of really amazing fossils, but oftentimes you really have to hunt to find them. As I like to explain to folks, it's actually fossilization, generally speaking, is a pretty rare process. The vast majority of living things don't become fossils. And oftentimes, even though we're finding lots of really amazing fossils, we're still only looking at like a tiny piece of an ecosystem of what all was living at any given time.
Yeah. And what I really like about trace fossils in particular is that you're instead of getting the creature as it died there, instead of getting the bones or the teeth, or a brachiopod that has likely been transported away and rapidly buried, trace fossils are preserved in place, and they preserve what that animal was doing when it was alive, which body fossils don't necessarily do. They can tell you more about the anatomy and things like that. So trace fossils to me are very interesting in that it helps you understand a little bit more about the paleoecology.
Yeah, I talk about that too sometimes because I love that about trace fossils. As I put it, I was like, body fossils will tell you like about what something looked like, but trace fossils can tell you how it acted, how it behaved when it was actually alive. And that's kind of, I think, mind-boggling to think about a little bit, that you can actually learn about a living thing that probably hasn't existed on Earth for hundreds of millions of years, and like how it moved or how it behaved when it was alive. And that's pretty amazing. So. Yeah,
that's definitely my favorite part about paleontology in general, is to be able to take your mind and put it back in time and imagine what was going on. Sometimes I come up with crazy theories like this animal probably had a little baby that was walking next to it according to these little tracks. I kind of come up with my own story, but it's really fun to imagine what could have been happening at that time.
Well, and that's the other thing about trace fossils, is that it really does invite imagination because, as you pointed out during the talk, I mean, oftentimes we don't know exactly what made trace fossils. We can hypothesize. We can take fossils and compare them to trace fossils and say, well, maybe it might have been made by this thing,
right.
But we don't always know for sure.
That's certainly the challenge, and that was the biggest challenge for my thesis.
Yeah, I'm sure.
I can understand that.
All right. Well, thanks for being here on National Fossil Day, and finishing out our day of paleontology talks here for the Geology Festival.
Of course, it's been an absolute pleasure. Thank you so much.
Absolutely. Thanks. And as you said, hopefully folks will have questions that need answering here
yes.
online on social media. So, all right. Thanks again.
All right. Thanks, Joel.
Well, hello again, everybody. We're back where we started here today for National Fossil Day, which is actually a really, really awesome fossil site right up here on the rim of Grand Canyon. So, of course, being up here on the rim, as you probably know from the talks that we just saw today, that means that we're in the Kaibab Formation. And paleontologist Ranger Ronnie here, what kind of fossils again are in the Kaibab Formation?
This Kaibab locality shows so much of the different kinds of shallow sea marine environment that's here. And it's really a snapshot in time of a really robust community that was here. We have all kinds of brachiopods and crinoids and sponges. This was a very diverse community at one time.
Absolutely, yeah. And a lot of the fossils in Grand Canyon, of course, are in layers way down inside the canyon. They're really hard to get down to and actually see some of those fossils. But these here in the Kaibab, up on the rim, a little bit easier to get to. So we actually have a dedicated ranger program under normal circumstances where we'll bring groups out to actually look at these fossils. But even though we do a guided ranger program out here, you're also free to come out here and look for them yourself as well.
Yeah, and the reason that we can be here and look at some of these is that these non-renewable resources, like everything else in the park, is protected. And the folks who have been here before us left things just the way they are. So come out, find them, photograph them, try to identify them, enjoy them, and leave them for the next people that come behind you.
Absolutely. We have over 6.5 million visitors coming to Grand Canyon every single year. If everybody took home a fossil with them, there would very quickly be none left for folks to come and see. So yeah, absolutely. Please come out here, look for fossils, enjoy them, take pictures of them. Just make sure to leave them where they are. So, thanks for joining us today for this day of GeoFest, celebrating National Fossil Day. Tomorrow's topic is all about time, geologic time. Which is a pretty cool concept. It's a little bit difficult for humans to understand. It's a concept that fossils actually tell us a lot about. But tomorrow we're going to be diving deep into geologic time and thinking about time and learning actually how geologists learn the ages of rocks. So I hope you'll join us for those talks tomorrow.
Sounds interesting.
Absolutely, yeah.
Yeah, well, happy National Fossil Day, Ranger Joel.
Happy National Fossil Day, Ranger Ronnie. Thanks for coming out here with me today.
You bet.
Night, everybody.